Well, hey, welcome everyone. I appreciate you coming to this review session. Um, it's hard to believe that we're talking about getting ready for an exam because I feel like the class just started. Maybe you feel that same way too. I wanted to give you an idea about what to expect for the exam and do a review session of the exam. But really, the main point of this is for you. So if you have questions, please feel free to chime in. That's my first priority. So you can either speak or type your questions. I'll be looking over at the text box to see. Let me just first ask, can you hear me okay and see me okay? Awesome. Great. And once again, thank you for attending. I really appreciate it. So tomorrow at this time, we'll be in the science building in room 140 for the exam. And I'm not sure if you are familiar with Mission College campus, but if you're not, you might want to spend a few extra minutes to go and check out. And actually, the building is really easy to find. It's right by the parking lot. You'll need to have photo ID with you, and also your study guide. You want to bring your study guide. Not that you can use it, but you'll be turning it in for points. And then also a Scantron and pencil. And the exam is 50 questions, and they're multiple choice, true, false, and matching. So matching, I mean like there's a series of possible answers and a series of statements, and so you have to match them with each other. So no fill in the blank. You might be happy about that. And so that's the format of the exam. And honestly, it usually doesn't take people very long to take the exam. Not that it's a race or anything, but people often can finish within a half hour. But you have 2 hours if you like. So that's just for you to know. And I'll be starting off our session asking you if you have questions, asking your classmates if you have questions. And so there's that opportunity as well. And I'll have nice big whiteboards to be able to answer your questions. So that's what's going on. You might have noticed that there's, let me say hello to Paula. You might have noticed that there's now extra credit available, and there's 5 different extra credits worth 10 points. So since our class is a 1,000-point class, that means that each of them is worth basically 1% of your grade. So it's possible to increase your grade by 5 percentage points through extra credit. It would take some time to do them all, but I'm saying that possibility is there. They're all due at the very last day of the class, July 26th. You can turn them in between now and then anytime. I don't recommend that you do them all at the last possible moment, of course, because you'll be working on your, studying for your exam. But anyway, they're available for you. So that being said, do you have anything for me? Do you have anything you want me to particularly focus on, or any questions from the study guide, or just any questions in general? So that's an open offer. So as I'm talking, if you think of something, you just let me know.
Hi. I think it's pretty straightforward. The slides are really good slides. So maybe just like a brief review on everything. So maybe more in depth, I guess.
That's what I've got for you. What I with the limited time that we have, I've got an hour. That it seems like a reasonable time for a review. So that's exactly what I'm going to do. I'm going to touch upon what I think are the most important points that you should know.
Mm-hmm.
If you're going through and that's what I want to understand better, than say, hey, can you explain that in a little more detail? And
For sure.
Any other comments or questions before we get started? Okay, cool. All right, let's go. We'll respond here. There we go. So I wanted to talk about the concept of, oh, hello, Vanessa. I want to talk about the concept of emergent properties because that's something really cool and unique about biological systems is that, well, you know, they're hierarchical. I'm sure you've got them memorized, the layers of hierarchy. With biological systems, as you go up the hierarchical level, then they tend to acquire more properties that they didn't have at the lower level. So that's called emergent properties. And so the example we talked about in class is like a food molecule. It can't really do much on its own. However, if it's a molecule that's working with a bunch of other molecules in the form of a cell that can actually move around and chase bacteria. And so having these properties of motion and reproduction are things that will arise as you go from the molecule level to the cellular level. But similarly, if you go from the level of individual to population to community, there are other qualities that will arise, other complexities that come. So that's a common theme, and it's called emergent properties. Wanted to make sure you know about natural selection. And so natural selection has resulted in organisms that are very well adapted for their particular set of circumstances in their environment. The example here being the moths. You know, in this environment, of course, this kind of moth is going to be recognized by birds and picked off and not survive as well as one that blends in better with the background bark. And so if then this moth is picked up at a greater rate, that means this moth will be able to survive, and surviving organisms are able to produce offspring. And whatever traits they have, they're passing on to their offspring. And so whatever allows an organism to survive better and longer and reproduce more, those are the traits, the qualities that it has that it's going to be passing to its offspring. And so the end result here is that the organisms that make it, the winners, are going to be very well adapted and have the traits that will enable them to survive better, because that's what they're doing, and they can pass that on to their offspring. There's a lot about the scientific method in chapter 1. Scientific method is something that we actually all do. It's just the trial and error of when we have run into a problem, the car won't start. What's wrong? Okay, well, maybe it needs gas. Let's try getting gas. So you will take an approach step by step, trying different things, those are called variables. And so it starts with an observation of the world and what's going on, and then from that coming up with a hypothesis or a guess on what might be happening, and then making a prediction. If I put gas in the car, then it should start, since if the problem was not having gas. So you make a prediction and then carry it out, testing it. That's the experiment part of it. And so in carrying out the scientific method, it's important that you will only change one thing at a time to be able to see its effect. The variable is anything that can influence a process. And so, you will be basically trying out each variable to see if it has an effect. And in the scientific method, in real life, if scientists find that something doesn't have an effect, then they're back to the drawing board, coming up with another possible hypothesis and checking that and looking for predictions of what might happen. And so in testing it, the experimentation process, it's important not to change too many things at one time. You just want to change one thing if you want to see what the effect of that thing is. Like, for example, we talked about this in class, looking at the effect of temperature on rice production. So you'd have a series of chambers that are similar in every other way. The humidity is the same, the amount of sunlight's the same, the soil that you use is the same, the type of rice you use is the same, everything's the same except one thing changed, that would be temperature. Then you can find out the effect of temperature. And so it's important to run controls, that is, keeping things constant, so you have a point of comparison. So control is very important and experiments. That's keeping things constant except for the variable that you're changing. And then any scientific experiment is going to require data. It has to be measurable. It has to be something that you can gather measurements on. And then with these measurements, then you can look to see if there's an effect and draw conclusions from that. So the cell theory, that is the idea that all living things come from cells, and that all living things are made of cells. And the gene theory refers to the fact that our traits, mainly were proteins and the macromolecules that we have, the qualities that then they bestow upon us. This information on how to be an individual, it's coded. That information is present in the DNA, and so it's the genetic information that really bestows upon us the qualities and traits that we have. And the DNA is in small packets that are called genes. And the theory of heredity is how this information can then get passed along in a population, generation after generation. And this information is passed as discrete units, the genes I was just referring to. And how they get passed along is very predictable. And we'll find out exactly why in the chapter on mitosis and meiosis. But there's a very logical and predictable way that we see this information get passed along. So that's the theory of heredity. And then the theory of evolution, it's, heredity, we pass traits from one generation to the next, but, given the long picture, over a long amount of time, we actually see descent is happening, yes, but there's also modification. And so, a population, the genetic makeup of a population will change over time. And that is, of course, due to natural selection, which is the mechanism for evolution. Those qualities and traits that are going to provide an advantage for an organism's survival will be the ones that are passed along. And so over time, we will see more and more and more traits that give a genetic advantage for that particular environment, which of course is also changing too. So those are the main points in chapter 1. If you came a little bit late, I made the offer, just jump in. Either you can, talk or type in. I'm looking on the sidebar all the time to see if you guys have any questions or if you want me to elaborate on anything, or if there's a study guide question that's related that you'd like me to talk about too. Oh, hello, I. So now chapter 2, now we have atoms and molecules. An atom is characterized by the number of protons it has. So that really identifies it. And a proton, of course, is a subatomic particle. And so if I say that there's an atom and it has 8 protons, you would know that it's oxygen. Not that you need to memorize this. I'm just saying that it's very distinctive. There's no other atom that has, let's see, 15 protons except for phosphorus. And so atoms with the same atomic number have the same chemical properties, and they're said to belong to the same element. Now, the mass number is the number of protons and the number of neutrons together, because those are the subatomic particles that have a mass, or you can think of it as a weight to them. And they're about equal in their mass. And so what happens then is, when if we look at the mass numbers are just about double what the atomic numbers are, because that would account for both the protons and the neutrons. Now, the electrons, of course, are another subatomic particle. They're negatively charged, but their mass is so much smaller than protons and neutrons that it doesn't even really doesn't really show up. It's just, it's negligible. And the reason these aren't exactly double, if you're wondering, I say they're about double, that has to do with the fact that there are isotopes that exist. We'll talk about that in a minute. So it's not always the case that the numbers of protons and neutrons are equal. And so that, difference sometimes will cause the mass numbers to be slightly different than double. But that's a general rule of thumb. So mass number is protons and the neutrons. Now an ion is an atom that has a charge to it. You can think of it as a full charge, and the reason it has a charge to it is because it either has an extra electron or electrons, or has lost electron or electrons. And the way atoms are is that the ideal is to have a filled outer valence shell. And so the first shell here for sodium is 2 because the first shell holds 2. The next shell holds 8 and has 8. The next shell has a capacity of 8 also, and it has 1. So what situation is sodium in? Well, it needs 7 more electrons to fill that outer shell. Well, that doesn't seem very doable. Wouldn't it be much easier if the sodium simply lost an electron? I'll block it right there. And then it would have a filled outer shell. So if it has 1 less electron, then that means it would have 11 protons, because that's what sodium is, 11 protons and 10 electrons. So protons positively charged, electrons negatively charged, then they're not equal. It's going to have more positive than negative charges, 1 in fact. And so that's why you see Na plus, the ion written as Na plus. It's possible some of them might lose, other atoms might lose 2 electrons, and so they'd have a couple of pluses, or some will gain an electron or 2, so they'll have 1 or 2 minuses on it. But ions you can recognize because they've got that superscript of a charge, and they are charged. Now, isotopes are atoms that have the same number of protons, like here, carbon. What makes carbon a carbon? 6 protons. These are all carbon. They have 6 protons. But they have a different number of neutrons. And all of these carbons exist. Now, this is the most common, having 6 protons and 6 neutrons. But there are also going to be carbon. There is carbon that exists that has 7 neutrons and even 8 neutrons. These aren't as stable, but they are out there in less numbers. They are out there and they exist. So one says that these are different isotopes of carbon. So isotopes, same number of protons, different number of neutrons in this case. So that's not going to affect the charge because neutrons are neutral. Back to ions now. Ions, being that they have a charge, will often do ionic bonding. So let's take a look here at the sodium. We already talked about sodium. The best situation would be for sodium to drop an electron. The chlorine atom here has a number of protons that has 2 in the first shell, 8 in the second shell, and then 7 in the third shell. So it has a situation of either losing 7 or gaining 1. Well, gaining 1 is much easier. And if there is sodium about, that's the perfect situation, right? Because sodium is looking to dump an electron so it can have a filled outer shell. And chlorine will gladly pick it up, which means that it would have an extra electron. And so it's going to have, you can see the superscript of negative. It's going to have a negative charge. Well, these then are oppositely charged, and opposite charges attract each other. And so the sodium and the chlorine ions then would have no reason to go far from each other. The opposites attract. And this is, of course, what builds salt and salt crystals. There's an attraction between the opposites. And that attraction is an ionic bond. Oh, hey, welcome, Alan. Now, covalent bonds are bonds that are more than just being attracted. They're actually connected. I guess, think of it as more connected to each other in a more stronger fashion, because the electrons are actually being shared by more than one atom. In the situation with oxygen, it has 8 protons, and so we've got, I'm sorry, it has, yes, it has 8 protons, 2 in the first shell, and then it has 6 in the next shell. To fill its outer shell, it would need to have 2 more. Where can I get those? Well, let's look at hydrogen. Hydrogen is an unusual situation in that hydrogen is simply a proton and an electron, a hydrogen atom. And so the proton and the electron here, it only has 1 in its outer shell. So to be able to fill its outer shell, which is the first shell, which only holds 2, it needs another electron. Well, it can form this nice companion relationship here with oxygen. And so if the electron, hydrogen's electron, is shared from 2 hydrogen atoms, in fact, then oxygen can have 8 in its outer shell. And similarly, the hydrogen ion would share one of the electrons from oxygen. And so then it would be able to have 2 in its outer shell, fulfilling that. And, and it works out. The covalent bonds are a stronger bond than ionic bonds because there's actual sharing between the atoms. Now, this sharing isn't very equal because in this situation here, we've got 8 positively charged protons, and this is 1 proton. Now, an electron that's negatively charged is going to be more attracted to where there's 8 protons than 1. And so the electron consequently is going to be spending more of its time, it'll be moving all around. It'll be spending more of its time by the oxygen than it is by the hydrogen. So it's being pulled that way. And so the significance here is the fact that the electron then would be not the positively charged proton and the negatively charged electron are it's going to be pulled more towards oxygen, leaving a partially positive charge here by the hydrogen atom, since the electron is spending more of its time here. So you have a question. Covalent bonds are always stronger than ionic bonds. That's what I thought was the case. Always, I'm not sure. I'm always afraid of that. I'm always afraid of the word always. So I would need to look into that, and I don't mind doing that. And I will be seeing you tomorrow. So if you would like to look it up too, that'd be great. And then we can get the answer to that. See, I want to say hello to Gloria and Tuan and Timothy, who have joined us, and let you all know that please, at any time, if you have any questions, I'm always checking this little text box over here. So just go ahead and type them in. So what that creates is a polar molecule. So water has poles to it. That is, it has a negative pole here. It's not a full negative charge like we saw in the ions, but it's a partial charge. And then the hydrogen ions, around the hydrogen atoms, it's going to be a partially positive charge. And so what that means is that water is polar. And so that's going to have some special qualities to it, meaning that if you have a bunch of water molecules together, then what's going to happen is that they are going to align themselves in a certain way. And so the positive part of the water molecule will be attracted to the negative part of this water molecule. And so, they'll align themselves and they will actually kind of cling together. And you see that with the in a water droplet, that cohesion quality that water has. And it takes quite a bit of heat energy to be able to separate them. And so that means that water is pretty stable in liquid form. and it takes a lot of energy to convert water from one state to the next. And what that also means is that on our planet, we can have water in 3 different states. We can have it in solid and liquid and gas because of this quality of water. So I'd like to talk a little bit about pH. Actually go back to the water molecule and discuss the fact that sometimes oxygen will win that tug of war, and it will gain the electron to itself. And so the hydroxide ion would be formed. And so it basically has an extra electron because it took it from hydrogen. So the electron, it would be an ion, OH ion, because it has an extra electron. And the hydrogen would be just the proton itself because it's lost its electron. So that would be H+. And so that happens. We have hydroxide and hydrogen ions that form. And in water, in liquid water that's neutral, most of the water molecules would be H2O, not shown in this diagram here, but most of them would be together, H2O. But every once in a while, you see some that are been pulled apart. And the number of hydrogen ions and the number of hydroxide ions are equal in, by definition, in a neutral solution, such as pure water. Now, of course, others exist too. And this gives you an idea of the amount of hydrogen ions and the hydrogen ion concentration. And because it's a logarithmic scale, and this refers to the superscript here. So the pH represents the amount of hydrogen ions, and having a smaller number indicates a higher concentration of hydrogen ions. And so as we go to smaller and smaller number, we get higher and higher concentrations, which means something is more and more acidic. And it also means that it has more hydrogen ions. And so now in basic solutions, this the concentration of hydrogen ions is actually getting smaller and smaller and smaller, represented here in this logarithmic scale. And the amount of hydrogen ions are less, so the amount of hydroxide ions that would be available would be greater, consequently. And so we'll have more and more hydroxide ions. And similarly, the more basic that you get, the more hydroxide ions would be there. And if you get to really strong bases, you know, exercise caution around them, because they can be, you know, extremely corrosive because they're so reactive. Okay, so that's acids and bases. I want to now, I guess we're moving on to chapter 3. And this has to do with organic molecules, in particular macromolecules. So macro meaning large, of course. And there are 4 groups of macromolecules shown here. And something that they have in common is that they're composed of smaller subunits. So these polymers, we call them polymers, are composed of monomers or subunits. They're not identical, but they're similar subunits that will string themselves, assemble themselves together to make the larger polymers. And so that's what our macromolecules do have in common. So we'll talk about proteins, and the polymer of protein is composed of the monomer amino acids. And amino acids, the sequence of amino acids is going to determine what the protein is going to be, you know, what it's going to look like. And in particular, when I'm talking about protein, what its shape is. The shape is so important. And so, you know, depending on the function, you know, is it, you know, to help clot blood, or, you know, for forming a cytoskeleton or hairs or enzymes, the amino acid sequence is going to affect how that protein folds and consequently its shape. And so one very important class of proteins are the enzymes, and shape is very important for them because it is the enzymes that have a little pocket for chemical reactants in a chemical process for them to maybe to join or break up. Anyway, it's the shape of the enzyme that enables chemical reactions to be speeded up, which is called catalysis. The next group of macromolecules, the nucleic acids. So this refers to DNA and RNA. And so they're both very long molecules, DNA being the longest. It's our code, it's the information. And it stores that information. The subunits of nucleic acids are nucleotides. And so with DNA, it's double-stranded. RNA, it's not single-stranded. And these are, the function is it's storage of information. Information that gets passed on to who knows how many generations, generation after generation. The RNA is involved in taking that information and using it to actually create molecules that bodies need, like proteins, but it's still involved in information. It's just like the working copy of the information. So carbohydrates, that's the group of macromolecules that is composed. The carbohydrates can be small or large, I guess. They can be simple and just formed out of it. A couple of rings here, or maybe even one ring, like for glucose. This is sucrose, the disaccharide. They tend to be sweet when they're small, or it can be a series of rings. And so these are the subunits being monosaccharides. And what we're looking at here is glycogen, and that would be a storage molecule. And so it's nice in the sense that each of these little, the monomers can be broken off and then used when required as a source of energy, but in the meantime be used as a source of storing energy. And plants do that too, except they do it in starch. So energy storage is one important function. And then structure is another one. Cellulose, what makes plants have their toughness, their firmness in their cell wall, is the molecule cellulose. And another structural protein would be chitin, which you can find in arthropods, like this lobster. You also find it in fungus, in insects. So chitin, a nice hard structural protein. And then there's lipids, a group of macromolecules. It's maybe not as big as the other macromolecules. It's not as macro. But we do find the pattern of subunits, in this case, fatty acids. We have 3 fatty acids here that are working together, joined by a glycerol to form this oil, or in this case, this fat. And one quality I want you to notice about this is they're showing you that the chemical composition of fatty acids is really simple. It's basically carbon and hydrogen, and because carbon shares nicely the electrons with hydrogen in its covalent bonds, we don't find polarity. And because of that, the lipids are considered non-polar, so they don't have a charge, and they prefer to be with others of their kind without charge. They prefer non-polar molecules. Polar molecules have a charge. They're attracted to molecules that have a charge. And so the lipids will just kind of, presented with water, would just kind of cling to themselves, as you know. So fats, oils, another class of lipids would be steroids. That's like estrogen, progesterol, cholesterol, which we see in cell membranes. So speaking of membranes, one of the important molecules in membranes, when I say membranes, this could be membrane outside of a nucleus, this could be membrane of an organelle, or it could be the cell membrane. There's all kinds of membranes in cells that makes the compartments. They're composed of phospholipids. So phospholipid would be one of these little things right here. where the circle there is meant to be the phosphate group, and it is actually polar, attached to two fatty acid chains. So phospholipids, you have a molecule that's like polar on this side and nonpolar on this side. And so the nonpolar parts, like I said, nonpolar refer to hang out with nonpolar. And so this automatically assembles in this manner because we have water, basically water being the majority of what you find outside and inside the cells. And so the lipids will keep themselves, and then that's nice because it makes a compartment. And you also find cholesterol embedded, and that is important in helping to give the phospholipid membrane a little bit of structure that it wouldn't have, a little more firmness. So there are two types of cells, prokaryotic and eukaryotic cells. So the bacteria and the archaea are called prokaryotic. They are single-celled. They're very small, and they don't have compartments inside. They don't have organelles. They do have DNA for sure. You know, they're living, but they don't have it surrounded by a membrane. And so they're far simpler and smaller than the eukaryotic cells, which do have all kinds of compartments for different functions. And that would consist of the fungi, the protists, the plants, and the animals are all considered eukaryotic. Anything multicellular is eukaryotic. Any living thing you can see with your eyes is eukaryotic. Now the bacteria are, like I said, small, but they still can carry out all of the qualities of life. And one of the things that living things have to do is make more proteins to because aside from water, we're mostly protein, and so that's done at the ribosome. So they do have ribosomes, and what happens, we'll learn in a future chapter, is that the genetic information from DNA will make a working copy, and that working copy, you actually can see it here. The RNA is going to direct these ribosomes on how to make proteins. So they do have ribosomes, and they do have DNA that's found in what's called a nucleoid region, and they have cell membrane, cell wall. Some might even have capsules on top of that, and some might have a flagella for movement with the flagella. Some will have pili, which are not for movement. Actually, kind of the opposite in the fact that pili are for attachment, and bacteria are. It's very common among bacteria to be able to see them use such a pili to bring two cells into direct contact with each other. The next step would be direct contact, and then the exchange of genetic information from one cell to another cell. So it's not sex per se, but it is a way for variation to arise among the prokaryotes. And so that is with the pili. So cell membrane, the cell membrane is fluid. You might not be able to get that from a static picture like this, and it might seem kind of strange that a compartment can be enclosed in something that's fluid, but just think of like maybe a bubble, and how the surface of the bubble can be moving around, but it still can contain the air. So it is a fluid, and it is contained by the phospholipid bilayer, which can also contain within it proteins, as you see here, proteins that go all the way through, or maybe proteins that are just on one end or the other. And attached onto the proteins, there might be carbohydrates that might be like indicators or markers inside this fluid. So because it's has a variety of, and there's cholesterol too, because there's a variety of molecules, the membrane. The structure of the membrane is called fluid mosaic. The mosaic combination. It's fluid and can move around. And so there's, of course, a lot of animal and plant cell structures, and I do recommend for this exam that you do get those, quiz yourself on that so you're comfortable on all of the different organelles. We're looking at an animal cell, and some things unique to an animal cell here are lysosome. That's a sac that has digestive enzymes for breaking down and getting nutrition out of something that an animal cell has taken in. And so we don't find this in plants because, as you know, plants make their own food. They don't ingest. So lysosomes are unique to animal cells. And there is a lack of a cell wall, you can see here. So I don't want to talk about each of the organelles here, but I do want to mention that there are some organelles that work together. So I'm taking a slice through right here. I'm going to be talking about what's called the endomembrane system. And so this indicates that these are connected in their functionality. I can imagine a situation where we have DNA, and from that DNA, RNA, the working copy is being made, and the RNA slides out through the nuclear membrane and makes its way over to the rough ER, where you can find ribosomes that are attached to it, and that RNA could then be read by the ribosomes to make a protein, of amino acids. And so that protein then can make its way through a vesicle that is basically being pinched off, because this is, again, phospholipid bilayer. And so we can have a part pinched off as a vesicle and then join the Golgi. And in the Golgi can then, that protein could be folded in a certain way or modified or something attached to it. And it could be finally prepared for its final intended use. And it could even then leave the cell through a vesicle and make its way to really any part of the body, if that's where it's intended, or maybe it's meant for some other part of the cell. And so because they're all connected, we say this is the endomembrane system. So that includes vesicles, Golgi, ER, lysosomes, which are sacs of digestive enzymes. The smooth ER is where you have the lipids and carbohydrates are made, whereas the rough ER is where proteins are made. So I want to talk about these two organelles because they're special in their structure. They look almost similar. They both have double membranes. They both have what's indicated in blue here, DNA. Yes, it's true. DNA is not just in the nucleus, but DNA can also be found in the chloroplasts and mitochondria and ribosomes. And this probably has to do with that their origin was probably prokaryotic. And then through an ingestion event, they then got incorporated into a cell and then became an organelle, and there's no turning back now. I can't just release them and expect them to survive. But anyway, they're more complicated than the other organelles. They're both involved in energy. Photosynthesis happens here. And then we'll see in chapter 7 that mitochondria is the powerhouse of the cell, and that's where ATP is made. And so they are unique organelles that have something in common. And the cytoskeleton it's interesting because the cytoskeleton gives structure, but it also is the part of the cell that allows movement too. So in this illustration here, you can see, well, that this mitochondria is not budging because of the matrix that the cytoskeleton, which is giving this cell its particular shape. It can't move from its particular spot. But the cytoskeleton also, we would see projections from cells sometimes as cilia or flagella. That's made of cytoskeleton. All the cytoskeleton components of the other 3 different kinds are all basically protein. They're made of protein. They're different types of protein subunits, but they're protein in nature. So when I say movement, we'll see in a later chapter, mitosis, that movement of chromosomes from one end of the cell to the other is because of pulling actions of parts of the cytoskeleton. It's called the spindle. And so it's both for structure and stability, but it's also for movement of the cell or movement of things within the cell. And then do know your plant cell parts too. Of course, the one that's really standing out there is the central vacuole. But I also want to point out the fact that we have a cell wall here made of cellulose, and there is connections between plant cells, plasmodesmata, that allow for passage of materials. And even though it's a plant cells do have mitochondria, where where it's important to carry out getting energy from glucose molecules. So the central vacuole is a place where the plants will store water because they don't have the ability to go and move around like animals do. So they just basically need to plan better, I guess, and store for leaner times. And so they'll store water. That also could be a place where sugars and ions and pigments are in as well. So once again, I just want to put my invitation out there. If you guys have any questions, I keep glancing over to see if there's anything in particular you want me to elaborate on, because this is your review session, and that's my highest priority. So I think we've made our way to chapter 5 now. And so I want to talk about energy and 2 categories of energy, kinetic and potential. So potential is stored energy in a cell. What we're talking about in cells, really. Okay, here's a question. When you mean to know the parts of the plant cell, you'll be asked to name the parts of the plant cell structure. I won't have a diagram there. In the exam, it would be, I think that'd be take place more like multiple choice or matching. This part, and this is a function. Can you match the part to the function? So that'd be the kind of thing I'd be looking for you to do. Does that answer your question? And then potential energy is the energy of motion. So kinetic energy would be like the movement of a cell or movement of things within a cell, something moving across the membrane. So chemical reactions go from reactants, or I also hear the word substrates, to products. And so this is a diagram to represent an endergonic reaction. And that is a reaction where the products have a higher energy level than the reactants do. And energy must be supplied in order for this endergonic reaction to go forward. Very common. If you want to make a more complicated molecule, you've got to put some energy into it to, get the components assembled. For example, making a protein would be considered an endergonic reaction from the amino acids. So exergonic reaction is one where more energy is released than energy is put into it. So the reactants or substrates have a higher energy level than the products do. So this might be example, breaking down glycogen and getting energy from that, energy being released, more energy being released, energy that will be used by cells. And so in this case, energy is released. But if you notice there's a hump on both of them in this example here, there's even though the exergonic reactions release energy, they don't tend to spontaneously happen. There's what's called an activation energy to be able to get the reactants to a product. And that's the case. Another example is, burning gasoline. You know that releases energy, but it doesn't just happen spontaneously. You need to have a certain level of heat or spark to get things going, for the combustion to occur. And so the activation energy sometimes is an impediment for the reaction going forward. And so what bodies can do, since we want to maintain a nice stable temperature, we don't want to ignite things, in order to get reactions going, they can be, the activation energy can be reduced significantly by having enzymes. So what an enzyme will do is it will just create an ideal situation. Okay, I'm sorry, there's a question about exothermic. Same as exothermic, exergonic. I believe so. I believe so. I would want to look it up so I would be absolutely certain, but I believe that is the same thing. Okay, and so in order to have the reactions happen without having to put too much energy into it, it is possible to speed that up by having an enzyme to act as a catalyzer of the reaction. And so here in this figure, you can see these substrates could possibly randomly hit each other at the right spot and form the right product. But given the little compartment that the enzyme has for each of these reactants, it just places them in the correct orientation. And there might even be a shift in the enzyme to get that chemical reaction going forward to create the product. And so it speeds it up. And they can be used again and again and again, enzymes. It might be that there's just one substrate and it breaks into two products. This is just an example. But, just moving the chemical reaction forward can be speeded up sometimes with an enzyme. Now, talking about energy, this is the energy molecule of this is the energy molecule of cells, ATP. And so ATP is adenosine triphosphate, and there's 3 phosphate molecules here. And the reason that it's a great energy currency for cells is because of these high energy bonds. And the reason we have high energy bonds is because phosphate has negative charge, and negative charges don't attract, right? They repel. And so keeping this bond together, there's a lot of potential here, a lot of potential energy, because if that bond is popped, which it is to make ADP, this phosphate can come off, it can release energy. And if that reaction of releasing this last phosphate is coupled with something else, that energy can then be used for that. So it's the endmost phosphate coming off releases energy. The cells can capture that then to do work. And so the question is, this allows us to use less energy. You're referring to enzymes? Yes. So having enzymes allows us to use less energy, where it might take this much energy to get the reactants together. With an enzyme, it would take this much energy. Okay, so making our way now to the last chapter, photosynthesis, which I think is one of the more complicated chapters. But I'll try and stress what it is that you really need to know here, because it'd be easy to get bogged down on the details. So photosynthesis is something that occurs in plants, of course, algae, And even in bacteria, there are photosynthetic bacteria. One thing you'll find in common with plants and the algae and the bacteria that carry out photosynthesis is they'll have some kind of color or pigment, not always green. And that will help in that first step, and that is to capture the light energy. So photosynthesis can be seen in 3 stages: capturing the light energy, that is photons, capturing and now using that light energy to make chemical energy molecules, and then using the energy molecules to build organic molecules and sugars. So really that's the goal of it, but we can't go direct. We have to have this intermediate. We need to change the light energy into something that could be used for the final step. And so the first step, capturing light energy, pigment molecules. Pigments will capture light energy. Wear a dark shirt on a hot day. You can figure that out real quick. The pigments will capture photons. And you find these in the chloroplast. And in the chloroplast, there are these stacks of sacs, like green pancakes to me or something. These are the thylakoids. And in the membrane of the thylakoid, so I'm right here, in the membrane of the thylakoid, you find these pigment molecules with protein. And together they form what are called photosystems. And so it's the photosystem that acts like an antenna to capture light energy. So light energy is abundant, but it's these plants that are able to use that and then convert the light energy into another type of energy molecule. How do they do it? How they do it is the photons will excite the photosystem. And remember how it's photosystem 2 first, because it's just the order they were discovered. We're just kind of stuck with it. So photosystem 2, the photon will excite one chlorophyll molecule so much that it will actually lose its electron. The electron will be released from that chlorophyll. And that empty spot doesn't stay empty for long. You can see in this diagram that even though an electron is being released from the chlorophyll, there's one that immediately is coming into and replacing that electron. It's being acquired from water. Water is required for the process of photosynthesis. This is why. And when water then donates an electron, what happens is that it will release oxygen gas and hydrogen ions. And so that is why the process of photosynthesis releases oxygen gas, which is great for us, of course, because we need it. So anyway, that's where the oxygen gas comes from. But back to our story of following the excited electron. The electron that got excited from a chlorophyll molecule, because the photon will drop in its energy level as it's passed from one of the carriers in the electron transport system to another. So it will drop in its energy level, and we're not just losing energy. What that is happening is that it is tied with hydrogen ions. So a hydrogen ion is basically the proton. And so the hydrogen ions will pass and make their way across the membrane into this thylakoid space. And as this happens, you can see that they start to accumulate. So electron dropping in its energy level, making its way eventually to photosystem 2. I'm going to pause there because I see that I have only 1 bar on my battery, and I would hate to lose you because of my computer. Speaking of energy, I still plugged up here. There. Okay, so the electron has made its way now to photosystem 1. What's happening in photosystem 1 is that, like photosystem 2, it also has an antenna type of situation where there's a lot of chlorophyll molecules, and a photon will come and excite one of the chlorophyll molecules and cause it to release an electron. Well, that electron, they can see, is quickly filled up from the electron that's been passing its way down through the electron transport chain. And so anyway, we're having a little bit of a swap-a-roo here. Bottom line is that we have now another electron that's being passed down the electron transport system, and will make its way finally to joining NADP+, and that is an electron carrier. And so electron carriers do just that. They carry electrons. And so it's going to pick up this electron. And when it does, it will also pick up a hydrogen ion and create this energy molecule called NADPH. So what's important here? Not knowing all of this, it's okay. Knowing that the result here is the production of this energy molecule, NADPH. And it has to do with photons exciting chlorophyll molecules. And in the end, what happens is that we produce this energetic molecule, it's an electron carrier, NADPH. Now, because we have had electrons passing down to lowered energy levels, we had hydrogen ions being sent to the thylakoid space. And we're getting an accumulation here of hydrogen ions, as you can see, and that's not a balance. Something's got to give. And so what happens is that the hydrogen ions that have been accumulating here inside these thylakoid membranes, inside the thylakoid space, will be allowed to go back into the stroma. That's the liquid. It will be allowed to go back into the stroma, but they are going to go back only through this enzyme called ATP synthase. Now, ATP synthase is an enzyme that will, from the kinetic energy of the hydrogen ions passing, cause a phosphate and ADP to join. And that's no small feat, because if you recall, ADP, phosphate, the phosphate and ADP is negatively charged. Having another negatively charged phosphate added, that requires energy to do that. Well, this is the enzyme to do that, and it's powered by the hydrogen ions passing. So we now have ATP. Okay, question. I thought the ATP was created in between photosystem 1 and 2. It's not. The position of these just happen to be next to each other in your book. I don't get concerned too much with, I guess, the where of it. But please know that it's, to answer your question, the hydrogen ions that are being passed in photosystem 2 from the stroma to the thylakoid space, it's because of the hydrogen ions being passed in photosystem 2 that we have the hydrogen ions accumulating. And because we have the hydrogen ions accumulating, wherever there is ATP synthase in the membrane, it will allow the ATP, the hydrogen ions to go through in the formation of ATP. So really, it's thanks to photosystem 2 that we have this accumulation. Does that help with your question? You don't have to worry so much about location. Okay, good. And so in the end, a lot of detail here. In the end, what have we done? We've made ATP. We've made NADPH. And good for all the oxygen-breathing animals out there. We've made oxygen, but that's a waste product for the plant, actually. So it's the ATP and the NADPH that allows the Calvin cycle to go forward. So this is the third part here. We've captured the light energy. That's the photosystems. We now have made ATP and NADPH, and now we're on part 3. We're now going to use it. And this requires the Calvin cycle and the input, the constant input of carbon, not a carbon-rich carbon. I guess before I go forward, let me answer Bill's question. Question about chemiosmosis. I understand that ATP synthase uses chemiosmosis. One of the quiz questions suggests that NADPH also uses chemiosmosis. I will have to maybe go offline with you on that, because I'm not sure which question it is. I would say NADPH does not use chemiosmosis. So let's talk more about that after I've stopped the recording or our one-on-one. Okay, so we have ATP and NADPH available. We've got plenty of carbon dioxide that's in the atmosphere. So now we have all the components. The components, it happens in a cycle. The Calvin cycle, actually there's several rotations of the cycle that happen before we get anything spit out here. And what we're making are the pieces that will come together and make glucose, the sugar. And so as the carbon dioxide enters, now carbon dioxide is not very energy rich. You can't, for example, you can't use it as food. It's not an energy rich molecule. The carbon dioxide will then enter into the cycle, and the carbons will become energized by joining molecules that are already present in the cycle. And the addition of phosphates from ATP will make energy rich carbon molecules, and now electrons being added from NADPH, which then as we go around the cycle, some of them will spin off every so often. Some will spin off to form glyceraldehyde 3-phosphate. Well, 2 of these can be joined to make a glucose. And so there's a lot of detail here for an intro class. Really what I'm looking for you, it's like listed here, that, you know, with the Calvin cycle, with carbon dioxide and with the energy molecules that we've just produced, it is possible then to create the components that will then be used to come together to form glucose and also other sugars. And it is a cycle, and so we do see the components of the cycle being reused again and again and again. Okay, so the purpose of the Calvin cycle there is to build sugar molecules. Yes, that is the purpose of the Calvin cycle. Absolutely. And it can't do that directly. We need to have the ATP and NADPH. It can't use photons, right? It needs those energy molecules, and so that's why we have multiple steps. Now, of course, a plant or photosynthetic bacteria are then going to use the glucose and get even more energy from that. But you'll learn all about that in the next chapter. And we still, and even though we're having a test, we're still proceeding forward. And this Thursday, I just want to remind you all that you still will need to work forward, and Chapter 7 quiz will be due by Thursday at midnight. So that's just something to keep in mind as you're studying. So that covers what I wanted to tell you. If you have any questions between here and there, please do write me. I'd be glad to help you. We'll have some time in the beginning of the exam too to answer your questions. And otherwise, I'm going to say thank you for your attendance. I really appreciate that. It's nice to have that interaction and see such interest in students. Looking forward to meeting you.